very well accomplished in science. She has published more than 45 peer-reviewed articles, and she completed her education from BSc AG and MSc AG from Andhra Pradesh Agriculture University and PhD from Indian Agriculture Research Institute in New Delhi. So she's working in the entomology sections, and so she has a lot of experiences working with SRI and their SRI's interaction with the insect pest management. So with that, I would like to request Dr. Padmavathi to take the lead for the talk today.
Thank you, Dr. Ram. Am I audible?
Yes.
Yeah.
Yes, you are.
Yeah. Thank you. Let me share my screen. Is it visible? Is it visible? My screen?
Yes, it is visible. Can you make in the slideshow?
Yeah. Just a second.
Sure.
Yeah. So, good evening, all of you. Namaskar from India. Thank you very much, Dr. Ram, for those nice introductory words. So I'll be speaking about insect diversity and dynamics in system of rice intensification, wherein we'll talk about my institute, Indian Institute of Rice Research, and rice area production and rice ecologies in India, and Sri in India, how it has entered into India and how it has spread, and also the evolution of SRA in India, and also the multidisciplinary team of Sri at IARR, and then followed by insect pest scenario in general in rice, what are the decadal changes, followed by seasonal profile of insect pests, and our studies in SRA, particularly at our research farm and also at multi-location study, and impact of this SRA on insect pests and factors responsible, followed by their impact on biodiversity, meta-analysis, and conclusions. So now coming to our institute, Indian Institute of Rice Research, as told by Dr. Ram, as it is one of the institute of Indian Council of Agriculture Research. And initially it was established in 1965 as All India Coordinated Rice Improvement Project. And in 1983, it was elevated as Directorate of Rice Research and with lead research in irrigated ecology as a mandate. And in December 2014, it was elevated to an institute that now it is Indian Institute of Rice Research with the main mandate like basic and strategic research for enhancing rice productivity under irrigated ecosystems and the coordination of multi-location testing to develop location-specific varieties and technologies for various ecosystems and dissemination of technologies. And as you have seen, our main mandate, this is irrigated ecology and also the All India Coordinated Rice Project. So this is one of the largest research networks devoted to single crop. We have around 45 funded centers wherein 196 scientists working. You can see here, these are all the 45 funded centers which are distributed across the country. In addition to those funded centers, we also have more than 100 voluntary centers wherein almost 100 scientists were working on this crop. And we also have around more than 30 private organizations with more than 80 scientists working on this rice crop. And if you see area and production wise, India stands first in the area in the world with approximately 47 million hectares, and second in production in respect to milled rice, approximately 137 metric tons, and first in exports, that is 16.35 metric tons. But if you see the productivity, almost it is rice yields, they was plateauing in the last decade because of many reasons, including the climate change that is happening at a very faster rate. Now, if you see the exports, 60 out of 16.35 metric tons, basmati we have around 5.24 and non-basmati 11.12, which we are exporting, and resulting in a foreign exchange of 86,194 crores of foreign exchange. And this is where the India, you can see how the export has increased. And if we see the projected demand and supply also, we still by 2050 also we'll be having some surplus to export this rice. And coming to the quantity and value of respect, value of this, as I told, it is around 86,000 we are getting from the foreign exchange because of these rice exports. And but still, if you see the population, population is increasing very fast in India. Almost it will be touching 1.62 billion in by 2050, and in 2030 now the the required production is around 130 million metric tons. But now we have already achieved this target by this 2025 itself. Now we are having 137 metric tons, but for 2050 we have to reach around 168 million metric tons, and that too keeping these challenges, particularly limited resources of land, water, labor shortage, and also the climate change impacts that are already showing very, very seriously in India. In addition to those challenges, we also have different rice ecosystems in rice in India. That too you can see here. Irrigated rice ecosystem is high with a higher area and also higher productivity, followed by rain-fed lowland production system. And then we have uplands, hills, below sea level also, and even the deep water also, where these are known as a sort of ecosystems where the productivity is not up to the mark. We can see here almost 1.2, 1.4 tons per hectare, which is low productivity, and still we have some opportunities to increase the productivity in these ecosystems so that we can achieve this required production. Now coming to the SRA introduction in India and also its spread. It was in 2002 when Dr. T.M. Tyagarajan of Tamil Nadu Agriculture University attended international conference on SRA in China that we came to know about this SRA. Particularly, he came to know about that SRA and he started experiments in Tamil Nadu Agriculture University with various farmers. In the same year, actually, Dr. Norman Uphoff, who's the lead person in SRA, he came to India and he gave a series of talks, particularly in Tamil Nadu, Andhra Pradesh, and also in NCAP at IRA New Delhi. And followed by this, a series of experiments were taken in all these states. And also in 2003, Dr. Satyanarayana and Jalpati Rao from Acharya Engineering Agriculture University, placed at Hyderabad, they visited Sri Lanka for an exposure visit, which was supported by Cornell University. And immediately after coming, Dr. Satyanarayana Garu was very much excited with that Sri experiment seeing at Sri Lanka, that he started 250 on-farm trials in 22 districts in Andhra Pradesh. And in the same year, there were many NGOs also started working on this SRA. You can see here in Karnataka also that spread was there. And then in Gujarat also in the next year, the Gujarat Anand Agriculture University also. In Pondicherry also, Pushpalata collaborated with them, and large-scale Sri experiment was taken on 4 acres. And then scaling up has taken place in Tamil Nadu. But the most successful SRA experiments were conducted in Andhra Pradesh, wherein a delegate from Bangladesh visited, and also a delegate from American delegation also visited to see. There was a cyclone in Andhra Pradesh at that time, and then when they saw that Sri crops, they withstood that cyclone when compared to the neighboring crop, which suffered the lodging. And this has really made the impact, and everyone started looking at SRA in a very big way. And in 2004 itself, actually, WWF team, it visited Angru and started collaborating with 250 farmers in 10 districts of Andhra Pradesh. And this is the first large-scale partnership of SRA in India. And slowly seeing all these successful attempts, Government of India also started advising farmers to adopt SRA wherever feasible, and it recommended also in Boro rice. There are many other NGOs like Pradhan, Crops, and many other CSA. All those NGOs also parallelly, they took up the SRA and resulted in the spread of this SRA in many states in India. And the most significant one I can say, that is in 2005, he is the Chief Minister of Andhra Pradesh and Agriculture Minister of Andhra Pradesh, wherein they went to harvest SRI field in one farmer, that is Sri Nagaratnam Naidu's farm, and seeing the yield, particularly in that SRI plot, immediately the Chief Minister announced a 4 crore program for popularizing the SRI method. And this was the impact that SRI created during that time. And you can see how it has spread to slowly to all the states and also all the districts here, wherever SRI was spread. Now, after that, the first decade, as I am discussing, 2000 to 2010, that was the reintroduction and validation across India in different states, followed by a series of meetings, actually, particularly in relation to policy dialogues, and also in relation to scaling up, and also in relation to what are the future lessons or future experience directions of this particular SRI. So there were many meetings conducted, and finally it was included in our National Food Security Mission, and it is taking place in that by the government organization, government itself. And in 2022, as you all know, in December, we conducted an international conference on system of crop intensification at Hyderabad, and most of you have attended that conference. So if you classify the spread and the evolution of this SRA in India, I can say that first decade was like this reintroduction and validation, 2000 to 2010, and 2010 to 20 spread and modified to suit the local conditions, and 20 onwards there was a sort of diversification with other alternative methods like direct seeded rice, AWD, natural and organic farming, particularly keeping in view the climate change scenarios. And now, with the after that, A. Satyanarayana Garu visited that Sri Lanka, and then he came to DRR and gave a lecture on SRI, and then WWF project, and also Dr. O.P. Rupela sir of microbiologist from ICAR campus. They were the persons who involved all our DRR scientists into a mega project. You can say multidisciplinary team was created, and all these you can see 12 to 14 members were there in that multidisciplinary team involving various disciplines. And I was the entomologist who was involved in that multidisciplinary team led by Dr. R. Mahendra Kumar, agronomist. You all know who's head at present and PI of ECRIP program. And you all know Dr. Mahendra Kumar, who has taken the lead on this project. And these are the results of that project only, what I'm presenting now. And followed by that, we conducted around 3 national symposiums in collaboration with the Society for Advancement of Rice Research and also WWF in 2006 at Hyderabad, 2007 at Agartala, Tripura, and 2008 at Coimbatore. Now, these are the few glimpses you can see. This is Dr. Norman Uphoff, who visited our SRI plot at ICRISAT in 2006, and Dr. Rupela sir here. And these are some of the NGOs, CSA and also Crops NGO, and he's Mr. Vinod Gaud, who was present from WWF. And we had a series of meetings also, along with farmers also, his Crops Lingay Garu, who was associated with our project. And these are the experiments that we have conducted at ICRISAT and also at IIRR farm also. A series of experiments were conducted for about 8 to 10 years continuously to see, and we evaluated various methods also and various varieties also under SRA and normal method of rice cultivation. Coming to today's topic, I can say in general, if you see the scenario of rice insect pests in rice crop, from 1965, the decadal changes, if you see, there were only 3 pests at that time. Now it has increased up to 20 pests, which are causing yield losses. Though there are more than 100 insect pests that are reported so far, but these are 20 pests which causes yield loss, ranging from 21 to 50%. And the factors responsible include the planting high-yielding varieties over an extensive area, growing varieties that do not possess resistance, and cultivating rice throughout the year, with including the enhanced use of fertilizers and also the pest resurgence and secondary pest outbreak because of indiscriminate use of pesticides is also the problem for this. Now, if you see the seasonal profile of these insect pests, here in India, we grow rice mainly in 2 seasons. There are also some areas where 3 crops are also grown, but mostly it is 2 seasons, that is wet season, which we call Kharif season, and dry season, which we call Rabi season. And I am showing here the profile of major insect pests. You can see the stem borer, which will start in nursery itself, and you can see up to the harvest. And galmid sometimes you can see in the nursery, and it also will go up to the panicle initiation stage. And leaf folder, it starts at tillering stage and it goes up to the maturity. And plant hoppers, which basically starts after at maximum tillering stage and goes up to the maturity. And in dry season, that is Rabi season, which extends from December to May, you can see again stem borer from nursery to the harvest, and leaf folder from maximum tillering stage to the harvest, and plant hoppers also from little bit like panicle initiation to the harvest. And these are the pests which are basically found. And with the introduction of these systems, particularly the cultivation systems, you can see that is from more water to the less water, what we are seeing here. We do have an ecology which is having deep water ecology, waterlogged condition, where case swarm and swarming caterpillar are a serious problem, followed by transplanted rice, direct seeding, saturated or SRI, aerobic. That is, there is reduction in less water, and this is the pest profile which we can see, plant hoppers and all, leaf folder, and then early season pests, and also particularly in aerobic rice, we can see soil-borne pests like nematodes and root aphids, which are in high numbers. Now, we'll see with the introduction of this SRA, how the cultivation system has impacted the pest incidents. So we know that there are 6 principles of SRA. I'll not go deep into these SRA principles and all, but in the end, we will discuss about this SRA principles vis-a-vis the IPM practices. Now, directly we'll go to the experiments that we conducted at the research farm, both research farms, that is at IARR campus and also at the ICAR campus. And particularly in the initial years, we have tried to see the insect pest scenario in SRA and normal method of rice cultivation systems. So we have taken up 2 methods, and also we have taken up 6 cultivars, which includes 2 hybrids and 4 inbreds, that is IR64 Krishnahamsa and all. And we continuously conducted these experiments for about 4 years. And we established a blog that is known as the SRI blog at Patancheru with the help of Dr. Rupela sir used to take care of that experiment actually, and followed by Dr. Mahendra Kumar sir, and in which we have taken all the parameters, insect parameters in those fields. And after that, in 2012 and 2013, we have pulled up all the varieties that are present, popular varieties that are grown, and then we evaluated for their performance in SRA and also normal method for about 2 years. So we'll see the pest incidence in this. You can see here SRA Krishnahamsa and normal Krishnahamsa, and SRA KRH2 and also normal KRH2. Because you all know what are the differences, particularly with respect to number of tillers, number of leaves, number of all other things. So, and also the maturity, how it is early and all. So I'm not going deep into the agronomical parameters. I will restrict to the entomological parameters only in this presentation. So if you see the occurrence of pests, sorry, particularly in nursery, because it is only 14-day seedlings that we plant in SRA, and whereas in case of normal method, it was almost 1 month nursery will be there. You can see almost 5 insect pests were observed in a nursery, whereas only thrips were observed in SRA. Whereas with respect to the pests in normal and SRA in the main field, there are almost same number of pests were observed. And in addition to that, we observed particularly a leaf mite infestation, that too in Swarna variety, not in other 2 varieties, but in Swarna variety, we observed very heavy infestation of the leaf mite. which we thought we should be cautious, particularly under SRA. And coming to the other pests, you can see the horror maggot, gallmage, BPH, stem borer damage, and these were all less in SRA or low in SRA as compared to the conventional method, except leaf folder, which is high in tree method as compared to the conventional method. And you can see this is not only our study, even the study of our cooperators also. This is the study by Ravi again. Ravi from Coimbatore, that location also, they also found a similar result, wherein these or most of the pests are low in SRA method. And in with respect to the cultivars, what I have told, we have seen that white ear head incidence. You can see here white ear head incidence. Almost it was low in tree method as compared to the normal method with respect to the all the cultivars except two. Only in KRH2 and Akshay-Dan, actually, the white ear heads are more in those two varieties, but in all the other cultivars, it's less in SRA method as compared to the conventional method. Now coming to the multi-location study, after doing it in our research farm, then we took that multi-location study under the All India Coordinated Research Project from 2008 to 2013. And these are the 8 locations where we have taken with the 2 main treatments, that is conventional and tree method in a split plot design, and 2 cultivars, 1 hybrid and 1 variety, which is popular at that location. And almost 4,000 square meter area was planted in these 2 cultivation methods, and 7 replications were maintained. So we'll go pest wise in this case, and because there are more non-entomologists also, I'll tell a little bit about each pest also. So coming to the plant hoppers first, in India, we have basically predominantly, I can say, brown plant hopper, that is Neelaparata lugens, and white back plant hopper, Sogatella versitera. These are the nymphs and the adults of brown plant hopper, and these are the nymphs and adults of white back plant hopper. Though the damage is almost same, but you find them in mixtures also in the field, and you find this WBPH in the initial stage, followed by domination of BPH, and again in the later stage, in some places it will be WBPH. And this is a highly migratory pest. It comes from some other place, and wherever field is there, then it will cause damage like this, which is known as a hopper burn. So if you see the brown plant hopper and the WBPH, that is BPH and WBPH numbers particularly, they are very, very low in SRA method, which is a real advantage actually, you can say. See, they're very low, not only in hybrid plots and variety plots, both these WBPH and BPH are very low in SRA plots as compared to the conventional plots. And you know why it is? Because in normal method, we have the stagnation of water and resulting in a high humidity, whereas that is not present in the tree method. With respect to the yellow stem borer, if we say about stem borer, we have around 5 to 6 species of stem borers, but the most predominant one is this yellow stem borer, Scyphophaga incertulus. You can see this egg mass female, and the damage is in terms of dead hurt and white ear damage. And across locations, if you see, the conventional method has more dead hurts as compared to the tree method. And similarly, whiteheads also, the conventional method has more white ear heads as compared to the SRA method. In few locations like Raipur and Warangal, almost the damage is similar in both the SRA and the conventional method. Now coming to the next pest, that is leaf folder Nephelocrocus medinalis. Here also we have around 4 species. That this medinalis is the most predominant one, and it is also known as leaf roller, wherein it rolls the leaf. And you can see the larva and adult, and this is the damage it caused in the SRI fields. And in 2 locations you can see there is very high damage of leaf folder in SRA plot as compared to the conventional plot. And even with respect to the hybrid and variety also, the variety has more damage at Pattambi, whereas it is almost similar at Warangal. Now coming to the gall-midge, which is an internal feeder actually. This gall-midge also, it was observed at Ragolu and Rajendra, 2 locations, and it's almost at par in the hybrid and variety also, it was at par. And In both the methods also, it's at par, except at Rajan Nagar, but this is, we consider it as low pest incidence only. And next is case worm. Case worm again, it's a very specific pest, particularly seen in flooded conditions are the places where the water is logging. And this is the larva which makes cases, and then it starts feeding on the leaves here. You can see, and this case worm was observed at two locations in our study. And at both the locations, actually, there's not much difference in the, though there is high infestation was observed. See, you can see almost 50 to 60% damaged leaves were observed with respect to case worm, but there's no difference in the conventional and SRI method, which is at par. In both, even at Patambi also, it was at par. It was at par even with respect to the cultivars also, both hybrid and variety also. And this you can see in here, I kept all the references also, not only our study. In addition to that, the similar studies who reported the similar reports are also I have kept here. They also reported that low pest infestation. And in SRA, what we observed is this early season pests, what we call like horror maggot, thrips, and all are really a serious problem. Sometimes it will cause. And here in this case, though, coral maggot damage was observed, varying between the locations. Actually, high damage was observed in Patambi and those locations. But with respect to the methods, you can see there's not much difference in the damage between the methods and also the between the cultivars. With respect to the thrips, thrips is again really a serious problem in SRA, particularly even though it is 14-day-old also, you can see the pest incidence in many of the locations we have observed. And here you can see the variety has more damage as compared to the hybrid, and SRA has more damage as compared to the conventional method in both variety and also in the hybrid. Now, coming to the grain yield, across locations, what we have observed is, across locations also, you can see with respect to the hybrid also, with respect to the variety also, grain yield was high in SRA method as compared to the conventional method. Now, to see what are the reasons for this low damage, and particularly we know morphologically, if you see the leaves of SRA, they are sometimes they'll be, particularly if you enter into the field and all, you can feel that roughness of the leaf and all. So we thought we'll see what are the biochemical parameters that might be responsible or that might be resulting in this low damage. So when we have analyzed these, we found that ash, hemicellulose, cellulose, and silica, these are high in SRA plants, the plants that are taken from SRA when compared to the conventional method. So we assume and we presume that these are the factors, because of the presence of these are the increase of these factors, that low damage might be there in SRA plot. And in addition to that, we also have done another study wherein we have applied a topical application of silica, and we observed that feeding will really affect this mandibular wear and tear. You can see here, this is with respect to the leaf folder what we have done. And we can see the mandibular wear and tear, and this might be one of the reason why there is low damage in SRA. And coming to the biodiversity, in both the SRA and conventional method, you can see the phytophages are less in SRA compared to the conventional method, and whereas the parasitoids and predators, they are more in SRA as compared to the conventional method. And these are the parasitoids and predators that we found. You can see here. And we also have, there are also other studies wherein they observed that the dragonflies, damselflies, and also wolf spiders and ladybird beetles, they were found high in SRA in both the varieties, Deku and CAU, particularly in Arunachal Pradesh here. And we have gone for diversity indices also. You can see the abundance of the insects here. It is 263 and 210 in conventional method, and number of species, and also the Shannon index and Wagner-Perker index, and also the Maglev index. You can see there are indices which show the evenness and particularly the diversity of these insects which are very high in SRA as compared to the conventional method. So in addition to this, we have done a meta-analysis, particularly to see whether the reports that are already published reports about pest incidence in SRA, whether there is any similarity or a difference. So for that, we have gone for the meta-analysis using Meta for R package and version 24. And we collected all the literature from Publish or Perish software, and we have that literature was sourced from all these sites. And then we calculated the standard mean difference, which is known as the effect size, and also the I-square value, particularly which represents the Q static. That is, it shows whether there is heterogeneity or not. And based on this, so these are the studies, actually various studies, independent studies, we call them as, these are the independent variables or independent studies. So for each one, we have different studies. And then we have seen this. Here you can see the standard difference in the mean. It is 5.67. It has come, and I-square value has come for 75%. That shows that variability in pest incidence with two methods of cultivation are more likely due to the real study differences, and it is not due to the error. So that is, heterogeneity is there. And in case of I-square value of white ear heads, it is metadata. It shows that 65% variation is there, and this is the standard mean difference of 9.07 is there. And with respect to the leaf folder damaged leaves, I-square value for leaf folder damaged leaves was 44%, which was almost this. All these have a very effect size of 3.86. That is, variation is mainly due to the cultivation method and not due to the chance actually. And also with respect to the grain yield also, the I-square value was 99.6%. Actually, it indicated only 0.4% is due to the chance, indicating that cultivation methods, yes, these variations, whatever we are seeing, are mainly due to the differences in the cultivation systems. It is not due to the chance or due to the particular location effect actually. So if you see the conclusions, we can conclude that comparative studies between the SRA and conventional method, it indicated that lower pest incidence in SRE method as compared to the conventional method. Even the multi-location studies have indicated that incidence of stem borer, plant hoppers, and gall midge was found low, while the incidence of leaf folder, holler maggot, and thrips was found high in SRE method than in the conventional method. But in all the locations, grain yield was found significantly high in SRE plots. And gild composition, when we have seen, higher number of natural enemies were found in SRA and lower number of pests also as compared to the conventional methods. And meta-analysis revealed that that whatever the effect size is because of the methods only, not because of chance. And this is all can say that SRE practices establish a micro-environment in a crop therapy that is less conducive. And because of that, and also complemented with the high silica content and also cellulose, hemicellulose, and ash, those contents also, and greater number of natural enemies in the plot, these might be resulting in the low pest incidence, resulting in the less crop loss in SRA. And if you see SRA versus IPM practices, if you see, actually, they are almost similar to IPM practices. Say whenever there is a high plant hopper population, the first thing we say is to go for the wider spacing. And actually, SRA method already has a wider spacing of 25 into 25 centimeter. That might be the main reason for the low incidence of plant hoppers in SRA method. Therein there is an increased aeration and exposed rice plants to more sunlight, resulting in reduced PPH. And the other practice, that is conovidar, that particularly it was suggested to run the conovidar at least 4 times at 10-day interval and all, resulting in greater soil aeration and also incorporation of weeds in the soil. And because of this, as you all know, that weeds, they act as alternate hosts for many of these major pests, and the removal of them and at regular intervals, they reduces the pest incidence. And moreover, one apprehension we really had initially when there were reports about more number of tillers, vigorous plant growth, particularly increase in the leaf size, particularly leaf length and leaf width also, we thought there will be increase in the pest population also. And most of them actually, they definitely attract the defoliators, but significant increase in number of tillers and leaves also, if you see in terms of percentage. suppose each plant is having 10 tillers with respect to normal method and 20 or 30 tillers with respect to SRI method, when we calculate in terms of percentage, definitely there will be low damage only. And these SRI plants, because of their healthy nature also, they are sufficiently able to compensate for loss of defoliation. And you can see they are reaping paddy farmers which harvest through SRI cultivation. In fact, such photos, there were many print media has brought, and many NGOs also have worked out. So with this, we'll see for the way forward will be actually, though we won't say that SRI is a panacea or it should be grown everywhere wherever rice is grown, but particularly we can focus on limited areas where there is a low water or the less limited water area, water stressed areas, we can recommend SRI. And also particularly keeping in mind these climate change impacts, particularly it's very effective in places where the cyclone and all is there. It was standing, it was not lodging. So it is again we can think of going in those places also. And but still we need in addition, in spite of all these advantages also, we do have several socioeconomic issues with that have to be resolved, and still lot of factors are responsible for continuation of this SRA or discontinuation of this SRA, which we may have to really focus on that. And though we know that there are 6 practices which are most important for that, but still only using 1 or 2 practices also farmers are able to get good yields. So those partial factor productivities, and if there is any synergy between the factors also have to be worked out, so that only the major factors, if the farmers are adopting also, they should get maximum impact. And training and extension systems have to be strengthened again if we have to go for the future directives. And with respect to the insect, I say that the focus should be on conservation of biodiversity, especially natural enemies in rice ecosystem. And as such, there is low pest incidence, so not much to really worry about that. And I thank all the directors, particularly Dr. Viratmat sir and all who were there initially when we started these experiments, followed by Dr. Ravindra Babu sir and the present director, Dr. R.M. Sundaram sir, who is very encouraging and very supportive for all of us. And also I thank all the heads of entomology department, starting with Dr. Pasalu sir, who was there when we started these experiments, and then followed by Dr. Katti sir, who was continuously giving us support throughout these studies. And I thank the most important person, Dr. R. Mahendra Kumar, head and PI of agronomy, without whom we wouldn't have been able to do all these experiments. Actually, he was the main pillar of this SRA method, particularly. Now also he is taking up the DSR method also in the same way. And Dr. Santosh Rathod, a statistician who joined our team actually, who has done all the meta-analysis and all, I thank him. And special thanks to Professor Norman, really for his constant encouragement and also giving so many tips for study. The first day when we met, he was talking about many things about the insect pests, which I did not expect it will come from him. Thank you very much, Dr. Professor Norman, and all the entomology cooperators who have taken the SRA and also the experiments, and all my colleagues at ICR and IARR who was constantly encouraging me for the work. Thank you. Thank you very much for your patient hearing. Thank you.
Okay, thank you, Dr. Padmavathi. So it's a very comprehensive and very important information you share about impact of SRI in insect damage and population. So now you see already like completed. Can you hear me?
Yeah.
Yeah. Okay. So we have a few minutes for the Q&A session. So I would like to open the forum for Q&A session. So if you have any questions, any queries to Dr. Padmavathi, you can just raise your hand and you can directly ask to her. Before that, you can introduce yourself so that you know who is who.
Good evening. That was a good presentation. My name is Hemanta Kumar Pamarthi. I am from Chennai, India. Well, I am an eternal student in agriculture. So I do undertake studies in agriculture both in India and in Southeast Asian countries like Laos, Cambodia, Vietnam, Thailand, etc. So I've undertaken a few studies there. I'm interested a little more in the slide where you are showing the pie diagram on the phyto stuff. Actually, I have requested for a repeat of that slide so that I could understand it a little better. What I found was that in the places where I was visiting, mostly the insects that were hurting were the thrips, the stem borer, and then the leaf roller. Whereas in India, it looks like we have more insects. So that was an interesting study. So I just thought, any specific reason? Is it because we are more endemic, attractive or something like that? I mean, some clarity on that, if it can be possible. Thank you.
But yes. As you said, even I also stressed that in SRI, thrips were more. Actually, stem borer, particularly white ear heads, we cannot say it is more, but it is at par with the conventional method. And of course, leaf folder was more. I was showing that in across locations also we got that. But the minute you want me to share 1 second, can I share again?
Sure, yeah.
Yeah, this one. Just this one.
Yes.
Hold on for a minute.
Yes.
Hold on for a
Yes.
minute, please.
See, here we have seen the number and then calculated the percentage. Can you understand? I can share the.
No. I don't need that. I understand this. I'm only trying to compare this with what I have done earlier. Okay, this is comparable. Thank you very much. That's all. That's all my this thing. Thank you.
Thank you.
Can I? Myself, Dr. R. Mahendra Kumar. I think Padmavathi already referred. Can I go ahead, sir?
Yes, you can. You can.
Okay, Padmavathi, it was a very excellent presentation and also totally covered all the aspects of insect dynamics. My only worry is how to upscale this technology in a large scale. That I think we are not able to do it for last a decade or so. But now I think something like can we tie up with some carbon credits, these practices which will also help us to methane reduction and those things. If we can really focus more on that one than carbon credits, so that farmers who are reducing their resources, but still they are able to get better yields, and also they are also compatible with climate resilience, then this technology should go in a very big way. That is only if there is anybody want to share their experiences of other countries. Narman, I'm able to say Narman, Guruji Namaskar. I think we are all your children of SRI, and still we are now finding out what are the solutions, so that we can take up this in a very big way. Even though I have shifted little bit towards the direct seeded rice, where we wanted to popularize, but SRA principles are being utilized in direct seeded rice, and so that I think we can help the farmer for carbon credits and additional benefits for the farmers. so that he will continue to do this SRA practices as per his convenience. Thank you, one and all. Once again, I congratulate CH Padmavathi for excellent presentation.
Thank you. Thank you.
Thank you.
So anyone has any comments on this?
I just want to say hello to Mr. Norman. It has been quite some time since I last met him. I last met him, I think, in Vietnam, Hanoi, and then later I met him in Thailand, in Bangkok, somewhere around 2018, 2017 and 2018. After that, I think I didn't meet. So hello, Mr. Norman.
Hello.
This is Hemanta, who worked with Abha Mishra, Dr. Abha Mishra.
Yes, I think I should respond to the last question. I have thought a lot about the carbon credit angle for SRI promotion. So far, I've not found it yet very persuasive because it's such a complex matter. And unfortunately, when you start getting money involved, these get very complicated very quickly, and in a lot of ways, they can go wrong. My own view all along has been that there are enough demonstrable benefits from SRI management of rice crops that the extra incentive of carbon credits should not be necessary to get it widely adopted. The water saving, the higher yield, the lower costs, the resistance to pest diseases, resistance to stress of drought and other climate change factors ought to be enough, but they need to be documented. And that's why I really welcome all of the members of the research network. All of you, thank you much for participating, that we can develop a sounder base. The arguments that have been raised against SRI are most of them really quite spurious, I'm afraid. The idea that SRI is more labor intensive. Well, first of all, this is by definition more intensive if you reduce your capital inputs. The question is for the farmers, does it take him more or less labor? And we just, last week, our colleagues at Oxford asked a AI service the question, does SRI in India require more or less labor? And so this, this search engine went through the literature. We said we wanted peer-reviewed, quality literature. And I can share with you, I was about to send this off to Kumar and others in India. The response was, first of all, very complete and very well written. I was impressed by that aspect of it. But one important is that, yes, initially it will be more labor intensive, but once the methods are learned, labor requirements come down. And so the SRI not only saves water and seed and capital, but also labor once it's understood and well practiced. So again, but come back, I think that I would, those who want to work for carbon credits, please do. I hope you're successful, but I think that we should not be basing our hopes or our strategy upon that factor. We should be look at the water saving, which can be better documented. We should look at the cost reductions and documenting these in ways, and then they need to be communicated to farmers. And again, I'm not so concerned whether initially every farmer accepts, but we get at least a few farmers who understand, who try it, and they can themselves then become local advocates, local champions. This is where we best succeeded, is with farmers themselves taking the lead once they become, well, very knowledgeable and proficient in this. I think our biggest bottleneck is really having adequate equipment, by making sure that farmers have access to adequate and appropriate equipment for SRI. My colleague Lucy Fisher has been working on this for many years. We have an equipment network, which anyone interested is welcome to join. There's been a lot of research done in India. Thank all those who did that. I think we're getting very workable economic transplanters for SRI specifications. we have ever better weeders. Also, it was mentioned there's something of a transition from transplanting to direct seeding, which I think is going to be a big boost for SRI. SRI doesn't require transplanting. It says if you transplant young seedlings, transplant them widely, pay much attention to getting the roots protected in the process and well established in the soil. But I think that work on the transition both to mechanization and to direct seeding is probably a good focus. But there's so many other things. I mean, I really loved Padmavati's data, but it always suggests to me why we have fewer phytophages, more predators in the field. Anyone in entomology has had a field day for research, trying to figure out now it's multifactorial, multi-causal. So it will not be simple research, but there should be research on how do you explain the differences in insect populations in SRI paddy fields versus conventional paddy fields. How much is humidity the factor? How much is the spacing and the sunlight a factor? We just don't know. We have good ideas. We can make plausible explanations, but we're not yet where we'd like to be. Anyway, I appreciate that so many people have joined this morning, and I really thank Padmavathi for a fine presentation. I hope we can keep these up and I can continue to learn more from what's out there in the field. Unfortunately, I can't travel anymore, so I have to rely upon Zoom and email for my communications.
Thank you, Norman, for the comments and suggestions. We really appreciate for your presence, and we all encouraged with your presence and all the support to us.
Yes.
So yeah, so Dr. Padmavathi, you have any more comments?
Hello, I have one.
Okay, please.
Me, Lucy. I just posted in there in our research database. We have in the keyword pests and diseases, we have 100 papers in our database. I put it into the chat. This includes all the papers that we found, not just the peer reviewed. But also I wanted to comment that I saw a paper in the Hindustan Times yesterday about using water credits for SRI. That was in the title, using water credits for SRI. So maybe that's water credits might turn out to be easier possibly than, or more difficult than carbon credits. Okay, that's it.
So anyone have any question more? So Dr. Padmavathi, I have a small query. So generally we observe like more stem borer if there is a less water in compared to the flooding one, right? I'm not an entomologist, but it's my general observation. So if there is less water stagnant, so you see like more stem borer infestation in rice.
No, I don't think it is with respect to the water, but with respect to the spacing, you can see a lot of difference.
Okay.
Because spacing is, see, sometimes what happens is the spacing is more and spacing is above 20 centimeter also, you can see. By chance, if there is a gap between the plants also, you can see more stem borer damage. But with respect to the humidity and water level, not much difference.
Okay, thank you.
Yeah.
So I think now is the time to take the picture. So I would like to request everyone to open the camera, and we have a Thomas over there who will help us to take the picture. Please open the camera. Thomas, are you there?
Yes, I'm here, Ram.
Okay, okay. Did you take the picture or need to wait for a while?
We can take picture now.
Okay, okay. Thank you.
Okay, everyone. We need to take a picture for a moment. At my count, in 3, 2, and 1. Okay, that's all. Thank you, everyone.
Okay, thank you, Thomas. So I guess Thomas has already shared the link for the participant certificate, right? So I would like to request everyone to fill the form.
Can we take it again because we missed a few?
Oh, okay. Yeah, we can take again.
They're doing it.
Thomas, do you want to do again?
Yes, yes.
I think we should. We missed Eiji in Japan and also Iraq.
Okay, everyone, at my count. Okay, in 3, 2, and 1.
Okay,
Boom.
do again.
Hello, Kadir.
Yes, thank you. So
Thank you,
I
everyone.
think we have 4 more minutes. If anyone have any comments, any questions, any queries, or any thought to share, are welcome.
I have one
Yes, please go ahead.
question is about the effect of the wider spacing. If there's wider spacing, then there's more airflow and more sunlight coming in at the same time.
Mhm.
Do those have a big effect on any of the insects that you were working with related to the spacing?
Yes.
I think
Yes.
you already answered some of this.
Yes, yes, Lucy. As you said, that wider spacing, actually, wider spacing is what we recommend even in our IPM practice also. Not to go for closer spacing, keep at least like this, because it is transplanting method, no? So we tell them to keep place like that so that there will be sufficient spacing between the seedlings. Closer spacing will really attract plant hoppers, both brown plant hopper and also white back plant hopper. Plant hoppers will be more if there is closer spacing, more tillers, because no aeration. If it is dark, then the adults will sit and start laying the eggs. That is how they spread from in the field.
It's
Because
the aeration, but is it also due to the airflow or the sunlight or what exactly with the
Yeah.
spacing is the cause of that?
Yes, yes, sunlight and airflow also. That is why we recommend to go for the alleys. Say we say that you remove one line after every, say, 2 meters like that. Then so that there will be airflow and aeration also will be there, and sunlight also will go. That is one reason. So that's what we will recommend them not to go for closer spacing, to keep optimum spacing. Actually, we should not say wider spacing. We should say optimum spacing. So it may not be exactly like SRI method of 25 into 25, but definitely not very close like 10 into 10 centimeter. We don't recommend.
I'd like to raise an interesting question, which is still puzzling me, whether the response to spacing, humidity, and other factors, the multiple factors, is linear or whether it's bimodal. It seems to me that what we've been doing in our past rice practice is trying to figure out optimal spacing for density, for dense planting, and maybe it's a different set of factors, different dynamics for optimal spacing. That if you're going to plant them close, plant them 10 by 10. If you're going to plant them wide, plant them 30 by 30. We don't know, but I think we still have some work to be done on the spacing variable, as Lucy says, it's airflow, it's sunlight, solar exposure, who knows what else. And as I said, this is a multifactorial situation. We're just taking one variable at a time. Probably doesn't give us very satisfactory or sufficient answers. One of the things which I've seen from some places is that farmers are planting certain kinds of flowers, maybe weeds, on the bungs to encourage the growth of predators. That will change the equation. We can say this is what happens if you do just SRI, this rice production, but maybe you combine that with some ecological thinking, some ecological practice, you can get even better results. Again, that's a very good area for research to be done, because we know about the work done at the Sipe in Africa to control the stem borer and maize, that planting certain crops in the margins or even interplanting certain crops will change the whole ecological dynamics in favor of pest control. So this is an area for research. Entomologists should roll up their sleeves.
Yes, yes. We have done those experiments, Dr. Upaf. We have done different spacings, and then we have assessed the pest population in different spacings also. And again, we are IRR, we are doing even ecological engineering also. We have done experiments with different crops and all.
Thank you, Norman, for the comments and reply. So I think Francesco has raised the hand. Hi, Francesco. Do you have any queries to ask or any comments?
Yes, I have a question. First, I want to say that it's really, really nice to see so many friends' face on this call. And I'm happy to ask a question. Actually, is very much aligned with what Norman just asked. I was curious actually if you experimented the effect of SRI in combination with other agroecological practices that could be maybe crop rotation, intercropping with rice, or strip croppings, whatever. Their effect on pest population. And also if some of them maybe have a negative effect, like for example, in upland rice, I heard that it's quite possible to do mulching with SRI, and if there is a negative effect of biomass mulching on pest population.
Yeah, in one of the study, actually, one of our senior has done mulching with straw mulching they have done, and the pest population and natural enemy population they have taken, and that study is there. But with respect to the crop rotation and all, our intercropping, no, we haven't done.
Okay, thank you.
Really nice to see all of you, Francisco and all. I think we saw you in 2022 when you visited that conference. Nice to see all of you.
Yeah, likewise. It's nice to reconnect after a while and to learn about all the work you're keep doing. So very, very nice.
Thank you. Thank you.
Thank you, everyone. So I think we reached the time limit. So I really appreciate for sharing very important information about the insect diversity and dynamics in system of rice intensification by Dr. Padmavathi. And there are a lot of good comments in the chat box you can see. And we are really grateful for sharing such a good information. So like we have more than 50 participants from around the world. This is a really successful webinar. And I really appreciate for your time, for listening, and for your passion to about SRI, and good to see all the good friends after a long time. And with that, I think we are going to close the webinar today. Thank you, everyone. Have a good night, good day, and good morning.
Thank you. Thanks a lot. Thank you for giving me this opportunity. Thank you very much.
Thanks.